Alright, so today we'll be looking into a portion of protein synthesis since we cannot look through the whole process, which would take way too long. I want to talk a little bit about transcription, which is the process where DNA makes RNA. Before I can continue further, I should probably mention a little about RNA and the three types. They are mRNA, tRNA, and rRNA. And I will talk about each when we get to that part of the process. So transcription as a whole includes some different processes of its own. It's a step-by-step process that starts off with initiation, moves on to elongation, and finishes with termination. Okay, so this is the nucleus, where all the DNA is contained. The DNA has all the codes for creating proteins to be used by the cell. These codes are groups of nitrogenous bases in the nucleotides of the DNA, the nucleotides being the subunits of DNA. Now, the DNA is in fact so important that it cannot leave the nucleus. It needs something to copy and send those codes to the processing units. This is where mRNA, or messenger RNA, makes its entrance. But first it must bind to the DNA. This is the stage of transcription called initiation. The portion of DNA we'll be looking at that is getting worked on by the RNA units is called the transcription unit. This is the transcription unit. A little enzyme called RNA polymerase recognizes and binds to DNA in the 5' to 3' direction at the promoter region of the DNA. The promoter defines when the transcription unit is supposed to begin. In this case, the RNA binds to the transcription initiation complex of the DNA strand. So the RNA binds here and continues to take the complementary base pairing of the DNA template. This is the elongation portion of transcription, where RNA polymerase simply adds nucleotides to the 3' end of a growing chain. The other end is the 5' end, but that is just where it all started. There are collections of proteins known as the transcription factors that are meant to recognize the key area of the promoter, also known as the TATA box, which shows the DNA when to start copying into RNA. It is a specific set of codes that tell just that. The codes are organized into triplet codes, or 3 nucleotides each. These codes also known as a codon. So an example of a codon would be AUU or CGU. The RNA polymerase moves along the DNA template, or the parent strand used to make the copy strand, and pairs every nitrogenous base with its counterpart, one of bases: adenine, guanine, cytosine, and uracil taking the place of thymine. As it is doing this, there must be a stopping point where the mRNA strand should cease to be made, and this is the termination end of the terminator. This is the point in the DNA sequence that tells the RNA polymerase when to detach from the template. So there is the finished RNA sequence, but is missing 2 things: the 5' cap, known as the guanine cap, and the poly A tail. These ends make it easier for the RNA to leave the nucleus. The whole end to the copying of DNA into RNA has its own name, and it's called the stage of termination. You would think with so many codes there would be a mistake or two, right? Well, the cell makes up for that using a process called RNA splicing, which is done by little molecules called spliceosomes. The spliceosomes edit the pre-RNA to make sure it is the correct sequence, pre-RNA meaning before it is modified. As if to confuse you further, the RNA before it is modified is also called the primary transcript. The spliceosomes are also made up of little molecules called snRNPs. This stands for small nuclear ribonuclear proteins. The parts that are edited out of the pre-RNA are called the introns, or intervening sequences. What is left are the exons, or the express sequences, that are carried out of the nucleus to the ribosomes. The ribosomes, in case you don't know already, are the protein synthesis complexes made up of RNA, which are the structural units of ribosomes. Ribosomes are located all over the cell, including around the ER and just floating in the cytoplasm of the cell. I should also probably mention quickly, the ribosomes are made up of two subunits. The first is a small ribosome unit which reads the mRNA codons. The second is the large ribosome unit which interprets and makes the codes into amino acids. Once the mRNA makes it to the ribosomes, translation occurs when another RNA type called tRNA starts decoding the mRNA and synthesizes the amino acids that form the polypeptide chains, which are the proteins. And there you have it. Granted, I only went in depth for one half of the protein synthesis, that being transcription. The other half is translation, which is the actual synthesis of the proteins. But for now, I will leave you at that. Thanks for watching.